Today we're going to talk about receptors and enzymes. So now that you have a feel for biochemistry and just what's made of what and what proteins are and all that kind of stuff, we can talk about receptors and enzymes. And this leads into drug discovery and drug approval, which is cogent now during this epidemic that we're having. So I'm going to go through this material. And as usual, I may or may not break it up into more than one set of slides. All right, so let's talk about all of these things. So drug design, most drugs, not all, most drugs work on proteins, and they somehow interfere with a biochemical process. So they can either interfere with it, meaning turning it down, they can turn it up, they can activate it, they can turn it off, anything in between. That means that for the first step of drug design, I would argue step 0 would be to understand the biochemical process. So if there's a disease process, how does that work? If it's a bacterium, how does it work? Where does it invade? With the COVID virus, for example, how does it get into cells? Those types, and how does it reproduce itself? So we talked about proteins before. They're polymers of amino acids that have a function. In general, there's connective tissue like muscle, but a big part of what proteins do is enzymes and receptors. So let's talk about each of these. All right, and just a little bit more review on protein structure. The function is very dependent on the structure. They're polymers of amino acids, they're huge molecules, all this stuff we talked about before. The disruption of structure is called denaturation. This is all stuff, for the most part, we talked about this in the biochemistry slides, right? This is an example. I showed you the same picture in the biochem slide. So this is protein. This is actually the NCC backbone. That doesn't show the R groups. You can see some of the R groups hanging off here, but this is hemoglobin. All right, so let's explain what enzymes are. Enzymes are biological catalysts. A catalyst, by definition, is something that speeds up a reaction without being consumed. So it itself is not used up, but it is important the reaction may or may not even happen. So this is just an example of a biochemical reaction. And this really would be better written for the purposes of this class as pyruvate plus NADH makes lactate plus NAD plus. This is an abbreviation for a molecule, so this is not a structure, it's an abbreviation. In case you care, it's nicotinamide adenine dinucleotide. It's derived from niacin, which is a B vitamin. This is the enzyme. It's called lactate dehydrogenase, and this enzyme allows this reaction to happen. If you look carefully, tell me what kind of a reaction this is. See if you can guess. See the double bond to oxygen and now a single bond to oxygen, gained hydrogen. See if you can guess for a moment. Also this, lost hydrogen, became more positive. Very good. This is an oxidation and this is a reduction. It's a redox reaction. All right, this reaction, just for fun, this is what happens when you overdo it exercise-wise and you start burning sugar in the absence of oxygen, and this is lactic acid or lactate. This is what makes your muscles sore after vigorous exercise. All right, enzymes are often involved in metabolic pathways. I showed you that before as well. There's a lot of things that go on biochemically that are not single reactions. This is a part of fat metabolism, but you can see there's literally 1, 2, 3, 4 different reactions, and this is just for one cycle. Each of these is controlled by an enzyme. All right. So if the enzyme doesn't work, this set of reactions doesn't take place. So that's what enzymes do. Now, how do they work? In general, the first thing that happens in an enzyme, and by the way, some terminology, the reactants in an enzyme reaction is actually referred to as the substrate. Then there's products. So there's a substrate, which is basically the reactant. So in this case, this reaction is this little half circle triangle becomes a half circle and a triangle separated. So the reaction is this substrate going to these products. But what really happens, the first step is a binding event. Where it binds, where the substrate binds to the enzyme, is referred to as the active site. So the first step is called a binding event. And this is where, if you remember the organic chemistry slides, stereospecificity comes into place. So this, you see how the shapes are complementary? So there's a binding event, now it's bound, then the reaction happens, and then it's released, and you can see how the enzyme doesn't get used up. It could be used over and over and over again. So enzyme plus substrate goes to the enzyme substrate complex, the reaction happens, enzyme plus product. So this binding event is very important because if this binding event doesn't happen, this doesn't take place. And this can be very specific. If this shape was changed a little bit, this wouldn't fit. All right, receptors are molecules typically on a cell surface. They can also be within a cell. And they bind to another molecule, just like the enzyme binds a substrate. The thing that binds to a receptor is called a ligand, but it's similar to a substrate, except it doesn't necessarily get reacted with, and it binds on the outside of a cell, causes changes on the inside of the cell. Analogous to, let's say you ring a doorbell. Right? If you ring a doorbell, you push a button on the outside of the house and a noise happens on the inside of the house. That would be the same type of thing. Or you put a key in a lock and it opens the door. Sometimes a molecule is brought in. Sometimes there's cellular signaling. How each receptor works, how does that signal get inside of the cell can vary. All right? This is an example. So here's a hormone, which could be a ligand, binding to a receptor. When it binds, it causes a change on the inside. And that causes a set of reactions to happen. So this, again, if I push the button on the outside of my house on the doorbell, it rings inside. When it rings inside, that causes my dogs to bark, right? Or causes me to come to the door. So something happens on the outside causes something to happen on the inside. So this is a receptor-ligand interactions are a big part of signaling. And if you remember back when we talked about the immune system, I mentioned that there was a whole bunch of cellular signaling that takes place. It's the same type of thing. If you look back in those slides, when you have the antigen presenting cell presenting the antigen, that is a receptor interaction as well. All right, so what do drugs do? Most drugs work on receptors or enzymes. And typically the problem is it's working too well or it's not working well enough. Okay, typically another thing is that, especially in a complex organism like a human, there's a lot of regulation. So some reactions are turned on when there's a signal that takes place, and some reactions don't always happen. There's a signal that turns them on, just like, again, ringing a doorbell. All right. So what if the signal is on and it's not supposed to? What if the reaction is happening uncontrollably? Or what if the reaction's not happening when the signal comes there? That would be a problem. All right. So typically drugs, that's what they do. They mimic the ligand. All right. Now, how do they bind? How do things bind? It's the lock and key hypothesis. A better analogy would be a hand and a glove. So you notice like a glove, if you wear a glove, the glove is similar to the shape of your hand, but when your hand goes into the glove changes the shape a little bit. That's actually a better analysis, but we'll just go with this. So these have complementary shapes, and the interactions have to be complementary. It's not only the shape, but if there's a positive charge right here, that means there'd have to be a negative charge right here. Right? So, and polar, non-polar interactions. So all of those types of things have to be complementary. So what we can do with a drug is we can make an inhibitor. So this would be an inhibitor, like a drug. And here's the enzyme, and here's the substrate. So what's supposed to happen is the substrate goes into the enzyme. But we make an inhibitor that binds to the enzyme instead. Now the substrate can't get in there. So that would be a way to prevent this from happening. Right, that's an example of an inhibitor. Right, so the drug can bind instead of the substrate. There are what are called, they can be antagonist or agonist. An antagonist prevents the substrate from binding and it blocks the response. All right, so the substrate can't get in, nothing can happen. Or the ligand can't get into the receptor, nothing can happen. But another way to do it is you could have the drug bind to the receptor or bind to the enzyme, and it still gives a response instead of the substrate, especially when it's a receptor. So the drug binds instead of the ligand, but the drug does the same thing that the ligand does. So it's a way of turning the switch on in the absence of the ligand. They can also bind to other sites that are called allo sites. They can activate, they can deactivate, they can attenuate. Just to give you a cartoon picture, so this would be the active site of the enzyme. This is an allo site. This is the substrate. So the substrate would go in here. If this binds, though, you see how binding here changes the shape. Now the substrate can't get in. So, and again, it can open to the active site, you can close the active site, you can turn it up, it can turn it down. These are just general concepts. Typically in a drug, there is some, it binds to a protein, causes a change in the protein that affects what the ligand is supposed to do or affects the process. What are the specifics? That's going to depend on the protein and all that kind of stuff. So, but in general, that's what happens. All right, I am actually going to stop this here and make a second video. So we'll stop here. Well, we'll stop here.